Welcome everybody. My name is Professor Nick Slevin. I'm a trustee of the Christie charity. I've been asked, first of all, to read out this housekeeping note. In the unlikely event of an emergency, 3 exit doors, 1, 2, and mine is down here. We congregate outside the building at the bike shed. Louise, not behind the bike shed, which is out through the doors on the left-hand side. Christie Charity, as you know, has had a series of spotlight events, and those are designed to showcase the research that's going on here, which we believe transforms the lives of Christie patients. So thank you for your support today. The focus today is on breast cancer. There are 60,000 new breast cancer cases a year in the UK. 80% of those are over the age of 50. What if you're under 50? What if you don't have access to screening? What if you're under 40 with a young family? So we're going to hear today from 3 speakers. We've got Professor Sacha Howell, who's leading the study, Dr. Hannah Harrison, who I've just met, and very importantly, we also have patient experience from Annette Illing. And welcome to her husband and her oldest daughter, Imogen, who I believe is skiving off school today. We're aiming to finish at 12. We'll take questions after the speakers. When I say 12, there's no extra time, certainly no penalties. So the speakers have promised me they'll keep to time. So obviously this study, which we're going to be talking about, the BCAN-RAY study, has been underpinned by the Christie Charity Sarah Harding Breast Cancer Appeal. So obviously, I'm sure Sacha will make some mention of that. So our first speaker, Sacha Howell. Thank you.
Thanks a lot, Nick, and thank you everybody for coming today. So breast cancer prevention research in Manchester is what I wanted to talk about. So yes, risk prediction and prevention both will go hand in hand. That's me, and this is Hannah. I'm not going to point her at her, otherwise I'll blind her. And she's going to come on after me, and then Annette is going to come and talk sense third. So why are we trying to prevent breast cancer? I thought it would be a nice simple slide, and they get progressively more complicated from here. This is why we want to prevent breast cancer. So this is the increase in the numbers of breast cancer cases that we have over the last 50 years or so in the UK. Some of that is due to screening on the left-hand side of this graph, but after about 1988, when screening came in, it's just carried on rising. So we are getting more breast cancer. Obesity is a big issue. Later first age at first pregnancy, reduced breastfeeding, and increased contraception and HRT use. So all of these things are feeding into that. And there may also be other things that we are as yet unclear on that are increasing the incidence. Nick was just talking, and in some respects I don't need to do much of an introduction because Nick has already said there is probably a bigger problem in younger women because we currently don't screen any women under the age of 50 who don't have a strong family history. And so on this graph, what I've just shown is breast cancer against bowel cancer against lung cancer. And I hope you can see that over here, in the 30 to 50-year-old age group, it's breast cancer that really takes off way before those other cancers. And that's probably due to the effect of female hormones on the normal breast through every menstrual cycle and also through pregnancies. But there is good news. It's not all doom and gloom, because when we look at survival, how well women do after a diagnosis of breast cancer, you can see that mortality, and that's really since screening was started in 1988, that falls off to about half of the levels that it was just under 40 years ago. Not only screening, but it's also due to vast improvements of the treatments that we've got. And even though this talk is much more to do with risk prediction and prevention, I did just want to put one slide up to highlight what the Christie charity does for breast cancer patients as well. It funds research fellows, and Nicole Brown is in the audience today. She's been with us for 2 years, has got 3 first author publications there and another 3 on the way. Yeah, excellent. 4, she says. And with one of those, by the way that we redesign treatment standards around the region, we managed to win a National Health Services Journal Award. We also did a study, or still are doing a study, looking at monitoring of breast cancer patients to see if we can reduce the number of scans that they needed. And the Christie charity stepped in to add additional analyses to those and funded a research associate. So this is just one example of what the Christie charity does and where all of your money goes when it's raised. Perhaps most excitingly for me now is this study, which is the bracelet study run by Nikki Floam, and that was used, the Christie charity money, the 2030 initiative that I'm sure you've all heard of, was used to fund a research practitioner. And that study is now open, and that is looking at how we might use breast milk to detect breast cancer early and to really help to refine risk prediction. So those are all things that the Christie is funding outside of the research that I'm about to present. This is the big issue that we still have, and that is that despite the fact that I've shown you that the mortality from breast cancer is falling, so the number of women in the whole country that are dying from breast cancer is reducing, the number of young women dying from breast cancer is still, unfortunately, too high. And breast cancer is the most common cause of death in young women of anything. So not just of any type of cancer, but of anything. And so that's why we really need to be thinking about prevention. So this is Sarah, super talented, vibrant, local lass, who, as I'm sure you're aware, was a member of the band Girls Aloud, who are pictured there on the right. Sarah was diagnosed with breast cancer, and I have to say that Sarah's mum, Mary, and best mate, Fran, are in the audience today, so thank you very much for coming. Sarah had no family history of breast cancer, was diagnosed aged 39. Unfortunately, it spread rapidly despite treatment, and she passed away after only 15 months. So that was utterly tragic. But what Sarah wanted to do was to support research and try and prevent other young women getting into the same situation that she was in. And this is where both Cancer Research UK and the Christie charity came along to fund the BCAN-RAY study. So BCAN-RAY stands for Breast Cancer Risk Assessment in Young Women, with the lighthouse really representing a beacon, and that's where we got that imagery from. And just very briefly, this is what we do within the study. So instead of just saying, well, if you've got a family history, you can come to a family history clinic, we know that the majority of women who develop breast cancer, even when they're young, don't have a family history. So this study, the women were invited from their GP, they scanned a QR code, logged on, and entered their risk factors. We sent them a saliva sample tube. They filled that, brought it to the Wythenshawe Hospital, had a low-dose mammogram, which was also important in their risk assessment. And those women that we identified as being at increased risk were invited to early screening and preventative therapy if they were keen to take that up. We've got a brilliant primary care research network in Greater Manchester. And the GP surgeries were extremely keen to get involved with this study, and we had 17 practices that were involved and recruited really well to the trial. 140 women out of the 750 that we recruited were at increased risk, and all of those women have accepted review, and I've reviewed them in the clinic. 60 have already started their mammograms, and 1 early breast cancer so far has been detected, and that was Annette. And I am incredibly grateful for Annette to come again, because every time Annette comes to this kind of meeting, it's reminding her of what has happened. So thank you very much, Annette, indeed, for coming. There was a lot of detailed acceptability work that went in, and I've just put one simple slide there. If you would like to talk about how acceptable this approach is to women, please talk to Sarah Heinmarch, who's sitting in the audience there. Did a PhD on the BCAN-RAY study, funded through the Manchester Cancer Research Centre, and is now a postdoc with us, looking at how we might do better in the future. In particular, having identified that the uptake for women, particularly black women, was really low. And we're worried about that because black women tend to get nasty breast cancers very young. So Sarah is now addressing that in the future. What we've also shown is that more than a third of women took up the offer of risk-reducing medication. That's really important. Most family history clinics around the country don't even offer it. Those that do get way below 10% uptake. So by doing this kind of approach, we think that we can actually increase the number of women who are going to accept that, and that will reduce the amount of breast cancer. And just to give you an idea, just using this, what's called an icon array, this is the approximate risk of the women that have come into the study and have been identified to be at increased risk. So about 24 out of 100 of them will at some point in their lifetime develop breast cancer. If we gave all of these women tamoxifen, it would now be 15 out of 100 that would develop breast cancer. So 9 women would not develop breast cancer who otherwise would have done and wouldn't have to go through everything that one does with a diagnosis. But I hope you see the same thing that I do when I look at those graphs, which is surely we can do better than that. And so why do those women, those 15, still develop breast cancer? And can we do better? Can we think of different approaches to reduce the risk more effectively? And so that's also what we're doing. And what we've done with the NIHR Biomedical Research Centre is set up a serial biopsy platform, asking these questions. Can we enhance response to existing agents like tamoxifen? Can we develop novel approaches for prevention? Can we understand more about how they work? This is what we do, and it's far too detailed for this talk, but effectively we take a biopsy of the breast, of one breast at the baseline, we treat for 3 months and then biopsy again here. And we do different imaging modalities, MRI scans here, we do mammograms as well for other studies. Now, ulipristal acetate is a case that I just want to discuss. That's an anti-progestin. So a lot of you will have heard of anti-estrogen treatments, but progesterone is actually the hormone that controls the breast, much more than estrogen does. So it's an anti-progestin treatment that we involved our first study. And I want to introduce Bruno Simões, who's a very talented Portuguese researcher. So we first employed him on the ulipristal grant in 2015. He did 3 years on that study, and then we went on to lead the other studies in the NIHR Biomedical Research Centre. That funding came to an end in 2024 when we had submitted the ulipristal study for publication, got a lot of positive comments back, but when we do that with a high impact factor journal, it means that we have a lot of modifications and what we call revisions to do. And that's when the Christie charity stepped in and said, but of course, he's great, we'll fund him for another year. They actually said, let's do 2. And I said, well, I think we'll just need 1. And so they funded Bruno with a nice bit of money that allowed us to do the experiments. And it's unfortunate for Bruno that he's Portuguese. Even more unfortunate for Bruno, his wife is Spanish. Anyway, we can talk about that later. And I'm very pleased to say that we then published the findings from this research in Nature in November. And yes, it takes a long time to do this kind of clinical research. So from the grant in 2014, it's taken us more than 10 years to get this into Nature. But this has created a huge amount of interest in the approach and how we might better analyse the samples. And now we've got 4 of these clinical trials that we've done looking at better ways of preventing breast cancer. And those 4 studies are here, one of which is ongoing. And what that has done is has allowed us to bring all of the samples, all of the data from tens of thousands of women involved in the epidemiological studies together in the Prevent Breast Cancer Research Unit. So yes, we're talking about the Christie charity, but we also work with other charities. We've discussed CR UK and Prevent Breast Cancer. We're all working together to try and solve these really difficult problems. And Bruno, I'm very pleased to say, is now the Senior Research Fellow on the Prevent Breast Cancer Research Unit, and we kicked off on the 1st of April. We did have a soft launch in September that was opened by a gentleman that you might know the name of, called Mr. Burnham. So the Sarah Harding appeal goes on. Her memory goes on. We had a gala celebrating Sarah's life down in London, which was a tremendous success. Ongoing fundraising from the charity team with doorstop campaigns that you may have seen. And thank you very much for the donations that have come in for that. And Girls Aloud reformed as a foursome and did an arena tour with £1 for every ticket coming to our research. And that was all facilitated by the charity, and I'm incredibly grateful for that. And it's that money that has gone directly into funding the Sarah Harding Young Women's Breast Cancer Fellowship. I am absolutely delighted, just in a second, don't stand up yet, to introduce Hannah Harrison, who is that fellow, and she's taking on that mantle, and she's going to show you what she's doing in the lab to try and make a difference. And what we would normally do in any kind of academic presentation is then thank everybody involved and put up a slide. The main group that I want to thank are the women that have been involved in these bits of research, because the vast majority of them, it is just altruism, sheer altruism. Yes, they're in the family history clinic, they're having their regular screening, but the biopsy studies, they don't need to do those. The treatment's not going to benefit them after only 3 months, but they just come, and we have women just willing and to come and do these studies all the time. And so it's a massive thanks to everybody who's involved in those studies from a patient perspective. But of course, this is a huge team effort, and there are lots of the team sitting in the audience over here. So thank you all, and to our collaborators as well around the world. So thank you very much, and I'll hand over now to Hannah.
Thank you, Sacha. Hi, everybody. So I was lucky enough recently to be awarded the Sarah Harding Young Women's Breast Cancer Fellowship to work here at the Manchester Cancer Research Centre. Today I'm just going to tell you a little bit about one of the aspects of the research that we've started to do in my team, and it's working towards the idea of a personalised way of preventing breast cancer. So kind of leading on from Sacha's great introduction, I think you all know that breast cancer isn't just simply one disease. There's not just a type of breast cancer that everybody gets. There's lots of different types, and that makes it quite difficult to treat. And it's really important that we remember that it's many different diseases. There's also many different risk factors that influence development of the disease and of those different types of breast cancer. It's extremely complicated. And as you saw from Sacha's talk with those prevention trials, one drug doesn't work for everybody, and it simply wouldn't in the cancer setting or in any setting, especially prevention. So we need to find better treatment options so that we can treat these individual women as individuals and treat their specific needs to help reduce their risk of breast cancer. ChatGPT made this picture. I think it kind of it sensed my anguish when I was thinking about the road from lab to clinic. I didn't ask it to say how hard it is, but it obviously knew from all of the other queries I've put in recently. But this road is really long. From us here in the lab, thinking of ideas, trying to develop these ideas, all the way to reaching the clinic and to having benefit. It's a really long way. And many, many treatments that we come up with that we think of fail before they even get out of the lab, really, or they get into kind of animal testing stages. And one of the main reasons for that, I would argue, is that we have a lack of any good models. We have a real glut of things that we can use to test these drugs and to answer our questions. So one of the things that we set out to do a few years ago, before the fellowship, was to develop a good model. And it was a model in which we could culture and we could look at human tissue as it should be. So this is, I'm taking you back to school now, and we've got some images from textbooks, basically. There's a breast, and if we zoom into it, within these lobular structures you can see here, it's a really tightly controlled, very specifically organized piece of tissue. It has fat cells around the outside, it has lots of different cell types around the gland, and then when you get into the gland, there's 2 layers of cells. The inner layer are the cells that produce the milk, and the outer layer are like muscle cells. They contract and they eject the milk from those cells into that duct, and it's pushed out to the nipple when breastfeeding. Now that tissue is extremely complicated, and it needs to be kept level throughout life, but every month, every cycle, as the hormones change through your cycle, that tissue will grow and expand, and then it will retract again if no pregnancy occurs or after breastfeeding. The problem we have in the lab is when we try to look at tissue like that, we generally, or historically, we take the tissue and we put enzymes on it and we break it into single cells. And we just end up with little plates filled with all those different cell types, but they're not representing the breast anymore. So we can't look at the really important interactions those cells have in the human body. It's really important that they're together, that they can talk to one another and influence one another. So we developed a model, and this is the first model of its kind, where we could take tissue that was donated following risk-reducing mastectomy, and we dissect the tissue into these tiny little cubes, a couple of millimeters in size. And then we put it, and this sounds really simple now, I'm telling you it like this, but it wasn't. We put it into this little well, and it holds the tissue inside this, it's a bit like jelly. Doesn't taste as good, but it holds the tissue perfectly. And what we see when we look at that tissue later, I mean, this is the perfect example of a piece of breast tissue, and it really matches that textbook image of a breast. And you can see here the duct where the milk's going to flow, and you can see those little lobules at the end with the 2 layers that produce milk and squeeze the milk out. So we've got this great model system where we can look at the breast and look at the changes in the cells. Another thing it needs to do is it needs to be able to respond like the human breast would respond in the woman. And one of the things that drives change in the breast is estrogen, is the hormones. And estrogen tells cells that they need to grow, that they need to divide, so there's more cells. So in that stages of the menstrual cycle, as the gland is expanding, it tells them to grow. So we can look at these cells in tissue, and the blue cells here are represented here. They're just sitting there, not really doing anything. If we add estrogen, those cells will begin to divide. And there's a marker we can use. We don't need to worry about that. When they're dividing, they go brown in the tissue here. And you can see, I hope, that there are 3 cells amongst this lobule in the breast that are proliferating. This is just normal tissue. We've taken it from the patient, and we see there's a little bit of proliferation, just jogging over. When we add estrogen, I hope you can appreciate that lots of the cells turn brown, lots of the cells begin to proliferate. So the tissue's responding as it should do in the patient to estrogen. That's vital for our research. And even more so if we add a drug like tamoxifen, which is an anti-estrogen, it blocks that. So we end up with less cells in this proliferative, in this growth state. So we have a tissue now that represents the breast, the cells are in the right place, all the cell types are still present, and it responds to hormones and drugs like we want it to. And the reason we're really interested in estrogen is because if we look at this old diagram I made years ago of all the different risk factors that lead to increased risk of breast cancer, quite a lot of them are really just a function of estrogen. So look at this one, for example. The longer your exposure, your lifetime exposure to estrogen is, the higher the risk. So the earlier periods begin to the later menopause is completed. More estrogen exposure, increased risk. So Sacha mentioned the ulipristal acetate trial, but another trial that he has carried out, and actually it's still ongoing, is a trial looking at the effect of tamoxifen on risk. And as he described, a sample is taken from a lady at high risk before treatment. She goes on to 3 months of tamoxifen, and then we take another sample and we look at that tissue to see how it's been influenced. And we use that stain I showed you to show proliferation, to show the effect of estrogen. We count how many brown cells there are. This is one of the metrics we use. And you can see in this data taken from that trial, a lot of the women, 50% of the women, we see a reduction in those brown cells. We see the tissue is not growing in response to estrogen anymore. But in others, about half of them, there's barely any change in the cells. They're just not responding. And we don't know why. Interestingly, when we put tissue into the explant model, the model I just showed you with the little cubes of tissue, we see the same. We see some of them respond and some of them don't respond. So a big question, and this is being carried out by a number of people in our lab, but also around Manchester, is to understand why this is happening. And can we pick out which women it will work for? So in the explant model, we're doing this by taking tissue from multiple women and treating it as an explant with tamoxifen and looking at identifying which women did what, which women responded how. I don't want to go into the details of how we're going to do that. What I really wanted to do was just acknowledge how lucky we are that we're in Manchester while we're doing this. Manchester is a centre of excellent research. It has the Christie Hospital next to us. But it's also got one of the largest networks of teams who work on breast cancer, and that's the Manchester Breast Centre, of which Sacha is the director. And it's made up of teams all around the city who are working for to improve the lives of people with breast cancer, to get better treatments, and a large number of us now on this kind of prevention angle. So this is just a photo of where we are here, and across the road in the Patterson building, Nightingale Family History Centre, and we have collaborators up on campus as well at the university who help us. So when we look and we find the differences and we develop new ideas for drugs, how do we test if they're going to work? We could do it how we've done it previously, as Sacha described with the clinical trial. Find patients who might be suitable, biopsy, treat them for 3 months, biopsy again and have a look. But as Sacha said, the paper that's just been published took, was it 15 years? Not that long. Felt like 15 years. It was quite long. It took a long time. But I think we can do it better, and this is what we're going to do largely. This is really the main focus of the funding that we've got from the Christie through Sarah's fund. We're going to use the explant model to take from a single patient. We'll make little explants, and we can treat that person, that sample, that patient with lots of different treatments. And then we can say which treatments does this patient respond to? And then we can start to ask why and whether we can work out how to personalise those treatments to our patients. So I've shown you a whistle-stop tour, and I'd love to talk to all of you about the model and about the ideas in more depth, so please get in touch. But I've just shown you today that we've developed this exquisitely fabulous model. I always say cool, and my husband says it's not cool because you're a scientist. So I tried to not say cool, but it's cool. And we can use it to understand individual patients' response and test new drugs in the future. The money from Sarah's fund has allowed me to start to develop my own team and to really drive my research forward. And I just wanted to show you who those people are. So Anthony is a student actually who's just writing up his thesis, and he's made some amazing advancements in the tamoxifen study. Khaled is a visiting fellow from Sudan who is also helping us in the prevention work. And the exciting thing is that we've got 3 new students starting in October, PhD students, who are all focusing on that question of how can we develop better preventatives and more targeted therapies for our patients. So I'm going to hand over now to Annette. And I said as we walked in, I don't know how to introduce you. I think Annette can introduce herself. I think, we've had a real privilege to meet Annette, and I've worked with her a few times now in the past. And as Sacha said, it's an absolute pleasure to have you, privileged to know you. Come up, Annette.
Okay, I'm going to use the clicker. There we go. So if you don't want to look at me, look at this. I was told it would be a good distraction. Okay, so hello everybody. I'm Annette. I'm 41. I'm married to Mark, and we have 3 daughters, aged 13, 11 and 8. And I work part-time as a teaching assistant at a specialist school. So I initially took part in the BCAN-RAY study in 2024, and I've also been a patient at the Christie. And I'm really honoured to be here today to give this patient perspective. If someone had told me a year ago that I would have appeared on the BBC and now be talking about my journey with cancer in front of all of you, I wouldn't have believed them. I'm one of those people that when they ask, how's life going? I'm kind of like, yeah, we're bumbling along. But that bumbling got a little bit harder for a little while. In the beginning of October 2024, I received a letter in the post inviting me to take part in the BCAN-RAY study. My initial reaction was, why not? I did speak to my dad and sister, who are both GPs, and we talked about the psychological aspect of knowing my risk score. I did a little bit of online research, saw some interviews with Dr. Howell, and I felt at that point that if I did receive an increased risk score, I would be offered lots of support, and actually now that it had been offered, I'd really like to know. So I submitted my online application and I was selected for the in-person appointment. At the appointment held at the Nightingale Centre, I had my low-dose mammogram and I gave my saliva sample. The process was really thorough. They went through everything. They talked me through the different pathways depending on what my risk outcome was. Timelines were discussed and I went on my merry way. In February 2025, I received my risk letter, outlining that I was at increased risk of getting breast cancer. I had a follow-up call with Dr. Howell in April, where he explained my results in more detail, and I had the opportunity to ask any questions. He offered me at that point yearly mammograms until the age of 50, and explained that I was eligible for the risk-reducing medication with hormone therapy. I came away from the call having lots of thoughts about my risk level and whether I actually wanted to start this early hormone therapy, but actually it was a decision that got taken out of my hands. As part of the BCAN-RAY study, I also took part in the additional research interview, where I answered questions about how it felt to take part, how it felt to get my risk score. And I do think that that's a really important part of the study, looking at how the psychological impact of knowing your initial risk score. So in June 2025, I had my first mammogram at the Nightingale Centre. And actually I was contacted within a couple of days because they had found some calcification on my imaging. And as they didn't have any other images to reference it against, I was called back. And at that point, I had a biopsy, an ultrasound, and another mammogram. 2 weeks after my initial mammogram, it was confirmed that I had DCIS, which is ductal carcinoma in situ, and that would require surgery. DCIS is early breast cancer, and it is when some cells in the lining of the ducts of breast tissue have started to turn into cancer cells. I remember asking the breast care nurse after we left the doctorate if I'd heard right. Did he really say cancer? I was given lots of information and I was told to contact them if I had any questions. It was a real shock. I thought, I am at increased risk, but that was maybe years away. And I think we just thought, it's the first mammogram, it won't be anything. My sister had prompted me the day before that I should bring my husband to the appointment, and I was very glad that he was with me there. And I remember messaging family and friends with links to DCIS information, trying to express that being caught super early, no need to panic, and bonus, free prescriptions for 5 years. Rightly or wrongly, over the next months, we just kept going, taking each step as it came. I think perhaps those first months were harder for my husband, having to watch me go through the surgeries, my recovery and other treatments, supporting me and keeping things normal for our daughters. We just kept our focus on the fact that my cancer had been caught so early, under incredible circumstances, and it was treatable. So at the end of July 2025, I had my first surgery, where they removed the tissue with the DCIS at Wythenshawe Hospital. It was a 4-week wait to get the results, and as it was the school holidays, we just carried on as normal. Although that week before I knew I was getting my results, I did start to get really, really anxious. But I knew at that point the worst case scenario would be that they would suggest another surgery if my margins weren't clear. So my results found high-grade DCIS with an incidental, tiny, grade 2 tumour invasive ductal cancer, which means that the cancer cells were rapidly dividing. Unfortunately, not all my margins were clear, which meant that some cancer cells were present at the edges of the tissue that had been removed, and it was recommended that I had a second surgery, and at that time also a sentinel node biopsy, which shows that if the cancer has spread from the original tumour to the lymph nodes. At the end of September, I headed for my second surgery, again at Wythenshawe Hospital. Another 4-week wait, and at the end of October, I got the news that the margins were clear, no spread to the sentinel nodes, and I was being referred for radiotherapy at the Christie. And at the end of November, I joined the Fast Forward Boost clinical trial, and I was randomised to the standard care group, which meant that I would have 15 sessions of radiotherapy, which we finished just before Christmas on Christmas Eve. And since January, I have been taking my hormone therapy tamoxifen, which I will aim to take for the next 5 years. Last week, I had my 12-month review with my breast care nurse. She was super positive, and like many other professionals I've spoken to, thought it was just incredible the way that my cancer had been picked up, and it made me ponder on the future calls that she might have. I imagine that currently it's a rarity for the breast care nurses to phone a woman whose cancer was caught so early and has such a positive outcome, and that with more research and studies into early detection and prevention, hopefully it will become the norm for these positive conversations for the breast care nurses. I feel like today is my opportunity to say thank you. Thank you to my family, friends and colleagues for all your support and keeping us going over the last 12 months. Being part of the BCAN-RAY study has literally changed my life. I now refer to the moment that I chose to take part as my sliding doors moment. If I hadn't have joined the study, my life would be following a completely different path, and I would be walking around not knowing that I had cancer growing inside my body. Being part of the BCAN-RAY study has also given me hope for the future. Hope in kind strangers out there donating their money, giving up their time to fundraise for the Christie charity and the Sarah Harding's appeal. I think when we give to charities, we don't necessarily think that we are going to be the ones ending up receiving that support, and we maybe don't recognise that by giving to charities like the Christie, we have the chance to change an individual's life. I have hope in research that these brilliant people here today and their teams who have dedicated their careers to gaining a greater understanding of breast cancer and looking at early detection and prevention. This research is vital for future generations and personally for myself and my daughters. I have hope that we live in a world where people still care about other people and want to support others. I feel I have received the very best care over the past year. The Christie itself is a strange and wonderful place in equal measure. Nobody really wants to be there, but we all share a common thread, going through treatment ourselves or supporting someone going through treatment. Everybody there, staff and patients, have shown me compassion and care. Smiles down those really long corridors, opportunities for little chats in waiting rooms. Everyone has their own story, but the setup of the Christie and all the little extras offered to patients and their families really goes above and beyond. My final thank you is to Sarah and her family and friends. In the midst of her own journey with cancer, she was thinking ahead. She was thinking about others, wanting to leave her own legacy. And it is a privilege that I am the first patient to have her life saved from this early screening research study that she so fiercely supported with Dr. Howell. So in closing, I would like to remind you of two things. One, your donations to charities like the Christie matter. And two, to check your breasts. And from now on, I probably will continue to say that I'm bumbling along when asked, but with the knowledge that my life has been forever changed.
I have to say, it's always very difficult not to get emotional when you hear patient testimony. So thank you, three speakers, fantastic talks. We do have the opportunity now for some questions. Any questions for the speakers?
Thank you very much to the speakers, first of all. I'm a breast cancer patient at the Christie at the moment. Obviously, I'm much older than yourself. But my cousin's daughter isn't, and she has also had breast cancer in Oxford. We wondered to what extent, and I've been asked to ask this question today from her as well, what impact genetics have. We both have the CHEK2 gene malfunction, and we wondered what impact research is having on looking at genetics and young people and older women as well. Thank you.
Yeah, it's a great question, and there are two aspects really to genetics. One is for what we call high risk or moderate to high risk gene mutations in genes like CHEK2. Other people in the audience will have heard of BRCA1, BRCA2, and so on. I think it's reasonable to assume that we will all know our genomes. And we will have the opportunity to know our genomes within the next 10, maybe 20 years. And part of that, we know that there's this national study looking at doing whole genome sequencing in neonates. And initially, it'll just be to look at the diseases that are relevant to very young people. But of course, once you've got the genome sequenced, you've got all of this information about CHEK2, BRCA1, BRCA2, and so on. And so what we need to do as a sort of society is to work out what we want from that. Do we want to get to 18 and then consent to release the rest of that information? And that will then undoubtedly identify many more women who carry these mutations, and then that can be used to help screening. The other thing, though, that we do, and what we did within the BCAN-RAY study, is to look at something called a polygenic risk score. And that is not looking for major gene mutations, it's just looking for subtle changes in the DNA. Individually, they either slightly increase or decrease the risk, but only by a tiny amount. But we're now looking for more than 300 of those. And so if you're somebody who's inherited many more of the ones that slightly increase the risk, then overall your risk will be increased. And that is the majority of those women at increased risk had a high polygenic score. That can also be obtained from the same whole genome sequencing. And so, as I said, in about 10 to 20 years, I think we will all know all of this information, and that will be guiding us as to how young women start to get their screening. This one size fits all, everybody gets 3 yearly screening from age 50, isn't good enough. And the National Screening Committee is looking at ways that it can change that over the next 5 to 10 years.
If breast cancer is in the family and you haven't got it, but your sisters have, what advice would you give to a woman who has a daughter who hasn't got it but may develop it? How they can't get through the door of the GP and get advice. They think you're balmy when you go, what are you fussing about? That is the first object, so, the first obstacle you hit is how do you get through the GP's door and how do you get your screening? Because I was very impressed with your courage in going for the screening, and I'm very touched by your testimony, but I think it's really difficult because I have friends and relatives with breast cancer, and how do they go about getting the screening for their offspring, etc. See, in a family, you can have 3 sisters, of which 2 have it, but not the third. What advice do you give the third? Thank you.
So there are already, there's guidance out there for GPs about who to refer into a family history clinic. But it is, we are still really working on guidance that first came in about 2004, well before any of the sort of mammographic density or polygenic scores were available. The polygenic scores are still not available on the NHS. So if you don't fulfill those criteria, then a GP will not refer that individual into the family history clinic. If those 2, what we call second degree relatives, so if that, I was going to use the jargonistic term, but I won't do it. If that woman's 2 aunts were diagnosed under the age of 60, then she would be eligible for a family history clinic review, as of course would her mother. So that is something that I'd be happy to talk to you about it afterwards. But I think this highlights the issue that this is an imperfect system that is currently just based on family history. And more than 2/3 of women, even young women, haven't got a family history when they're diagnosed with breast cancer. So that's why we're doing this study.
Thank you. I used to be a nurse at the Christie, and then since I left, I've developed breast cancer myself. So I've had treatment as well as been an employee there. So I just want to say thank you so much, because it's just a wonderful thing that you're doing. My sister, the reason why I stepped away from the Christie was because my sister developed breast cancer whilst I worked there as a nurse, so it all felt a little bit too close to home. And then once I'd left, I developed it as well. What my point is, I've got a daughter and I've got 2 nieces, and one of the nieces and my daughter are very anxious. We don't really know where to go for support, like health advice for them. They're still quite young, but they're worried about what might happen in the future, and we don't really have a direction to they're a bit lost, if you know what I mean. So,
yeah, that's a really powerful testimony. Thank you for that. And when we're talking about the charities working together, Prevent Breast Cancer is a really good place to go, as is Breast Cancer Now. So that both of those charities offer that kind of advice. And support. It is probably suboptimal because, in the NHS there is, we do rely on charities to try and pick up some of the pieces that the NHS can't really fulfill. But there is certainly a lot of advice from Prevent Breast Cancer that you could access to help. And know that we can't really talk about specific cases, but with that kind of family history, there very much will be early screening. So when people are over 18, they can get referred in to family history clinics. I would tend to say leave it a bit longer, because we wouldn't do anything for several years after that. But mid to late 20s would be a good time.
It's probably about 10 questions in one, but I suppose I'm thinking about how long will the funding be able to go for with your research, and how important it is for that funding to keep coming in, and then what's sort of the medium to long term goal? What do you want really to come out of this at the moment?
Good question.
Finally relinquished the microphone. This is like lab meeting. So if we're talking specifically the Sarah Harding breast cancer appeal, for now obviously there's a lot more funding than just that. That funds me for 5 years to perform this research, to pay my salary, and to give me the stability within the university to build a team. But for all of the people I bring on, I need to bring in money. And the funding landscape is a challenging one. We've got lots of grants that we're applying for to bring people in. We've got 3 students coming in October. But the landscape of funding is difficult. So having support from somebody like the Christie, which is a good block of money, a good block of time, means that we can make a really big impact and we can really kick this research off Your second question was sort of ultimate goals. I think for me the ultimate goal is to prove. The first goal is to prove this model is suitable and we can test lots of different therapies in it. The ultimate goal will be to get those treatments into clinical trial and see how effective they can be.
And in addition to that is to take the BCAN-RAY study forward to a much larger regional study where we try and implement that. The screening committee won't change what we do unless we demonstrate that what we do is feasible. And so we have to then go from right at the beginning, so getting people through, getting women like Annette through for their risk assessment, all the way through into delivery of the screening, effectively outside of the family history clinics, which is where we're currently screening the women in BCAN-RAY. So setting up separate screening hubs, delivering that screening and looking at how much and doing the health economic analysis so we can work out that it's actually feasible to do that within the NHS. And we're working with the National Screening Committee to see exactly what they would need for us to then be able to do the study and then deliver that. That's going to be a multi-million-pound study, as you might imagine. I'd also just like to acknowledge the university have also seen the value of this work, and beyond the 5 years for Hannah, she will then become a university employee and continue her research. So this is a team effort, and the university are part of that, and we're very grateful for the Division of Cancer Sciences for that promise.
I think Sacha mentioned earlier that the Christie charity are committed to giving Christie clinical research 30 million up until 2030. I think what we always look for more than anything is a sense of vision from the clinical researchers, and obviously the breast team do that par excellence. So well done on that. Well, perhaps we can wrap things up and show our appreciation for 3 fantastic speeches today. Thank you.